13. Virendra.
Hello, Pratik.
Good morning, sir. How are you?
Fine. So how are you now there?
Nice to see you, sir. Been a
Yeah,
long
yeah.
time.
Yeah, long time. So how the work is going on there? Are you working on COVID?
Not exactly. Earlier, when I was at Takeda, they were working on cell therapy. But a few of my colleagues are in Pfizer, and they have been working on COVID these days.
Okay. What is the new project now?
Sorry?
Any new projects? Any new projects are there with you?
New projects? Yeah, I'm currently working on a few related to melanomas these days.
In Takeda, what are this ones works you have done then?
Sorry?
Any developments in the earlier company which you have worked now?
Yeah.
In there, any analytical development you have done or with respect to we have vaccines only?
Takeda. In the previous company, I was working on 3 vaccine candidates for dengue, norovirus, and Zika vaccine. So mostly in the analytical development and stability platforms for those candidates. So they are currently in phase 3 for the dengue vaccine candidate, which is expected to launch next year.
Then you have left out that dermal, whatever the delivery system of powder, but you have totally switched over to the vaccines. But your PhD work was there in the dermal, no? Transdermal.
Yeah. Initially, I started with transdermal drug delivery, which further moved on to vaccine delivery through the skin. So I did a few projects during the PhD program, which included vaccine projects through the skin. So we were trying to develop something which is painless and needleless to deliver these vaccines.
Whether in the form of powder or in the form of liquid?
These are lyophilized powder vaccines.
Lyophilized powder, yeah. Yes. Okay, we'll start the session. I'll just have the introduction with you, about you. Okay.
Sure.
Good morning, delegates. Today we have got another one from the USA, our own alumni of the KLE, Dr. Pratik Thakkar. He has I will just read it. At present, he is working as an analytical development scientist, Birmingham site-led Replimune incubation in the USA. Earlier, he was working with the Takeda pharmaceutical vaccines business in the same USA. At present, he is working in the USA with the analytical development scientist. And just brief on, his major specialization is a transdermal delivery system, mainly with respect to the vaccine formulations. That is what he has done in the PhD also. Before that one, he did his UG and PG in the KLE College of Pharmacy. And when he was in PG, he was my student. And he was from 2008 to 2014. Then he was awarded from the UG and PG the best outgoing student from the KLE. And he has got a VGST grant. That is, I was also good guide for that. VGST grant, he got it from the Karnataka government, and he has got 5 publications, which are almost all in the Journal of Control Release, all the publications, and he has presented his research paper in the presentation of 2. And with this brief introduction, whatever the Pratik Thakkar is going to deliver on the culture of the cell culture therapy, that is with you. Thank you. Thank you, delegates. Pratik Thakkar, once again, and just I will hand over to you, the mic. You can share your slides.
Absolutely. Thank you so much for the kind introduction, Professor Bolmal, and good morning to everyone. Thanks for being here. I'll just quickly share my slide deck. Are you guys able to see my screen now?
Yeah.
Awesome. All right. So welcome, everyone. Today, I'll be talking a bit about gene-based cell therapy. And thanks to Keele University's College of Pharmacy for inviting me for this presentation here. A little bit briefly, I'll talk about my experience. So right after my master's program with Dr. Right after my master's program at KLE, I joined University of Rhode Island in Rhode Island, US, wherein I was working on transdermal drug and vaccine delivery for small molecules and vaccines such as HIV vaccines, the commercial HIV vaccine as well. I developed a small molecule patch that could sustain the delivery of powder drugs in high doses for up to 7 days. This was a laser-based system. I have briefly worked on microneedle vaccination as well. So these are painless, needleless vaccine candidates that can be incorporated via the skin. So these are causing lesser sharp wastes. Um, the needle phobia that happens in younger population is surpassed, and there is no need for cold storage of these vaccines because they are stable at room temperatures for a longer duration of time. So right after my grad school in 2019, I joined Takeda Pharmaceuticals in Cambridge in their vaccines business unit. And soon after, I was working on 3 vaccine projects, which are in different phases of clinical trials, mostly related to dengue vaccine, Zika, and norovirus. I was conducting viral titer measurements, including cell-based assays for these infectivity assays, ELISAs, plate-based ELISAs, and certain drug product stability and lyophilization studies for container closures for multi-dose vaccines for dengue, because in the Asia Pacific region, they have the biggest market. And I was managing these projects for the parent company that is in Japan. Currently, I'm working on oncolytic viral therapies at a company called Replimune. It's a mid-stage startup that started off with a team of around 50 people who developed the first oncolytic viral therapy in US that was approved in 2015 under Amgen's category of companies. And then they later moved on to form their own company at Replimune. So currently, a brief snapshot. So I was recently transitioned from Takeda to this company, not even 2 months now. And I'm responsible for setting up a fully functional analytical development lab here in the Framingham site, which is in Massachusetts. And I'm also responsible for supporting the GMP manufacturing with regards to biomarker testing requirements for their daily harvest samples for the products based on viral vectors that are being produced on site. So manufacturing unit is in the same building as I am working in currently these days. And apart from these, I'm leading tech transfers from the main parent company side, which is in Oxford, UK, and bringing these methods on to US, supporting 3 different candidates currently. We have, like the company has named them RP1, 2, and 3 subsequently. Apart from that, supporting GMP and process development groups with in-process analytical testing for drug product samples. And apart from these, the most important part is developing new analytical methods to characterize these new pipeline of candidates for their viral load, infectivity, concentrations, and other parameters. And eventually, I would also be involved with IND filings with respect to analytical methods and data generated through these methodologies. So today I'll be briefly talking about cell therapy as in general, gene therapy, various vectors that are used in these therapies, with a focus on immuno-oncolytic viral therapy, which is a recent hot topic in cancer research. and how these are applicable to analytical development fields and future directions in these fields. So starting off with cell therapy, which is also known as cellular therapy, cell transplantation, or cytotherapy, wherein viable cells are injected, grafted, or implanted into a patient in order to effectuate a medicinal effect. These could be transplanted from same species, from the same patient being a donor, or from a different patient being a donor. And these days, research is being conducted on trying interspecies cell therapy transfers successfully, but they are still under research. Historically, this all started in 1931 with Paul Nehans, who has also been called the inventor of cell therapy, who attempted to cure a patient by injecting material from calf embryos. And he claimed to have treated many patients for cancer using this particular technique, though they have not been validated by research. So it lasts back to about 90 years from today. So within these almost 190 to 100 years, we have come a long way. So various kinds of strategies are employed for cell therapy based on the species, as I mentioned earlier. So these could be allogenic, autologous, or xenogenic. Allogenic, wherein the cell donor is a different person than that of the recipient of the cells. And current research is going on for manufacturing products for certain conditions such as Crohn's disease, which is an autoimmune disorder, as well as other variety of vascular conditions. With regards to autologous cell therapy, cells are transplanted from a patient's own tissues. So disease cells could be taken up from certain bone marrow tissues, adipose tissues, or peripheral blood. They're extracted and then they're transplanted back after engineering to the site of injury or stress into the muscle or the cartilage. This could also involve isolation of matured cells from diseased tissues. So these can be later reimplanted along the neighboring tissues, and certain clinical trials for spinal injuries are ongoing for these conditions. And with the autologous cell therapy, there is limited concern for immunogenic response or transplant rejection, being the patient being the same donor himself. As mentioned earlier, for xenogenic cell therapy, the recipient receives cells from other species. For example, transplanting pig-derived cells to humans. Currently, it is only under research where human-derived cells are being transplanted to experimental animal models for the assessment of their safety and efficacy. Talking about gene therapy, a treatment that uses genetic material with the goal of changing the course of a disease. So the diseases are targeted by one or more of the following approaches, wherein a new gene can be added, or we could edit existing genes by either disruption or inactivation of that particular gene, or by correction, which means insertion of a new gene to correct those defects. Applications for these products last back to 1989. So between 1989 and 2018, over 2,900 clinical trials were conducted, with more than half of them in phase 1. And interestingly, a few products that were the first FDA approved ones for gene therapy were brought to the market. Spark Therapeutics launched their product Luxturna, and Novartis came up with Chimeria. Since then, Novartis came with another product, Zolgensma, and Alnilam came up with Patisiran, which have also received these approvals from the FDA. I'll go over the conditions or the indications that these products cover. So for the product from Spark Therapeutics, Luxturna, it is indicated for patients with inherited retinal disease. So an intraretinal injection of approximately 0.3 mL is given, and this is an adeno-associated virus-based gene therapy. The Chimeria product from Novartis is for B-cell precursor acute lymphoblastic leukemia, or ALL. Zolgensma, again from Novartis, is for pediatric patients that are less than 2 years of age. This came in recently, and for spinal muscular atrophy. This has been, I think, by far the most expensive gene therapy known till date. And Alnylam came up with patisiran, which treats polyneuropathy, which is damage to multiple nerves in adults. So gene therapy uses the various techniques of gene editing, wherein genetic engineering is involved, and in which DNA is inserted or modified in some way or replaced in the genome of a living organism. So these are targeted to site-specific locations, unlike older techniques which were randomly inserting genetic material into a host gene. So gene editing utilizes various technologies. I'll go over a few, although it's not my area of expertise. I'm listing out a few which the tools have been used in research, including zinc finger nucleases, a technique called TALEN, transcription activator like effector nucleases. A very hot topic these days about CRISPR, clustered, regularly interspaced, joint palindromic repeats. There's base editing using nucleobase modifications and meganucleases. So these certain techniques could be employed to edit out a diseased gene and insert a corrected gene, thereby utilizing the whole, correcting the whole genome, and then inserting these back into the patient. Could be locally at the site specific or systemically. Moving on to certain vectors that are used in gene therapy. These could be viral vectors or non-viral in nature. And the major vectors here in research that I have come across in terms of viral vectors, viruses are introduced into the genetic material of the host cell, and this tricks the host cellular machinery into using those as blueprints for viral proteins. So a number of viruses have been used for human gene therapy, including retroviruses, adenoviruses, HSV or herpes simplex virus. That's something I'm working on at Replimune. Vaccinia and AAV, those are adeno-associated viruses. Non-viral methods would include injection of naked DNA, electroporation, such as gene gun, sonoporation, magnetoffection, or use of nanoparticles. A certain level of advantage comes along with non-viral methods in terms of large-scale production and low host immunogenicity. But this initially produces a lower level of gene expression, thereby lower therapeutic efficacy. So newer technologies are able to overcome these challenges in the future. So talking about how this happens, this gene therapy could happen by direct delivery, wherein a therapeutic gene is packaged into a delivery vehicle such as the retrovirus, and it is directly injected into a patient, as you can see on the left side. On the other hand, cell-based delivery could involve a therapeutic gene wherein either adult stem cells are taken from a patient and genetically modified into a delivery vehicle, again, as a retrovirus and modified ex vivo. And then these are reintroduced into the patient. This could also happen another way wherein the embryonic stem cells are genetically modified and then differentiated in vitro, coming along with the therapeutic gene and reinserted again into the patient's systemic circulation. Certain disadvantages or drawbacks could include a harmful immune reaction, complications from inserted genetic material, adverse events, and unexpected gene activation or inactivation. Another hot area these days talks about CAR therapy, which involves, which is a combination of cell therapy, gene therapy, and immunotherapy. So in CAR T cell therapy, T cells from a patient are extracted and genetically modified to express a CAR, which is chimeric receptor, antigen receptor, and these cells are expanded and infused using this CAR engineered T cells. And so this infused target would bind to the target and kill these cancer cells. So on the right, we can see a process wherein blood is removed from the patient to get these T cells. And CAR T cells are produced in the lab wherein the inserted gene for CAR is inserted through this T cell. And once it has been differentiated, these can be replicated into millions of cells and then infused back into the patient, wherein it attacks the cancer cell, and which could lead to improvement in the condition, specific condition. Certain issues with CAR T cell therapy would include a limited number of available antigens currently, which can be targeted in the long term. And the major disadvantage for these kinds of therapy is the fact that solid tumors are extremely difficult to reach and treat. So T cells cannot go and easily attack solid tumors because of the environment. The microenvironment created by the tumor is so harmful, and the local conditions do not let the T cells to enter and inhibit the cancer tissue there. Moving on. Something that I'm currently involved with is immuno-oncolytic viruses. It's a combinational approach. Really, virotherapy has come up along in the last 5 years, wherein a virus is genetically modified to replicate only in tumor. The advantage of such therapy includes not only just attacking cells that are harmful or tumor-like, and not attacking the healthy cells in the tissue or surrounding tissue. Examples of that therapy would include herpes simplex virus-based therapy, adenovirus, or measles. Taking an example of a pathogenic virus, we all know that it's a parasite which is obviously harmful. So in the figure on the left, explaining or identifying as a virus, we genetically could insert an immune stimulating gene inside in its genetic material, and at the same time we can remove disease causing genes, which is for the selective targeting of tumors. So once the pathogenic virus has been stripped off its disease causing gene and inserted with immune stimulating gene, this can now be modified for use in terms of attacking just the tumor cells and preventing it from harming the healthy cells. In this image, the red cells signify the tumor cells and the blue ones signify the healthy tissue. So a direct targeting effect of virus, which will only happen in these red cells, would mean a greater chance of survival and better efficacy of treating this kind of tumors. So talking about the anti-tumor mechanism of oncolytic viruses, again, as previously mentioned, if this attacks the healthy cell, although it differentiates a bit, but at the end of the process, the healthy cell still remains undamaged. Whereas when an oncolytic virus enters a tumor environment, it will replicate and then leads to cell lysis within the tumor, which could further cause local inflammation, causing the tumor microenvironment being disrupted. Release of virus progeny, and this infects more tumor cells rather than the healthy tissue. And as an added advantage, it releases tumor antigens, which causes an anti-tumor immune response. This further inhibits the growth of the tumor and causes cell lysis at that particular tumor site. This is an excerpt from a paper from Dr. Praveen Bomareddy, who is currently the associate director at the company I work with currently. And he was involved with the first immuno-oncolytic virus program that was commercially available, known as TVAC or Imlygic from Amgen. And interestingly, he talks about oncolytic viruses being a new class of agents that likely work through two distinct mechanisms. One would be direct killing of tumor cells, and the second with an induction of an immune response against the injected tumor. So the second induction is an added advantage in this whole process. So the first oncolytic virus, TVEC, abbreviated as TVEC, or the Talimogene Laher parapVEC. It's a modified herpes simplex virus that was recently approved for the treatment of melanoma in the US, Australia, and Europe. These are patients in which first-line therapy was not successful. They had gone through more than 2 to 3 clinical trials for drugs for anti-PD-1 type therapies, which were not at all successful, and later ended up in the clinical trial for TVEC. In terms of safety, oncolytic viruses have an excellent safety profile. These can be genetically manipulated to prevent toxicity, enhance immune response, and promote tumor rejection. And at the same time, oncolytic viruses can be combined with immunotherapy, either in terms of a secondary injection or chemotherapy, targeted therapy, or radiation. So in a way, it's a collaborative approach to treat the tumor cell locally by injection, as well as other external sources of radiation as well. And currently, the local and systemic barriers to oncolytic viruses are not completely understood, and predictive biomarkers to identify susceptible patients are high priorities for future research. So that's the TVEC, first FDA-approved oncolytic therapy by Amgen for specific grade melanomas. So this is indicated for local treatment of unresectable or unreachable cutaneous, subcutaneous, and nodal lesions in patients with melanoma recurrent after initial surgery. These are genetically modified HSV type 1 products. And this produces an immunostimulatory protein known as GM-CSF. GM-CSF is a granulocyte macrophage colony stimulating factor, which causes cell lysis. This ruptures the tumors and promotes anti-tumor immune response. So currently at Replimune, we have 3 products ongoing, wherein RP1, as discussed before, is an oncolytic immunotherapy armed with GM-CSF and another protein known as GALV-GPR, or gibbon ape leukemia virus. This is in phase 1 for cutaneous squamous cell carcinomas for RP1 monotherapy. And we are trying combinational therapies with Opdivo, another company's product, for, as well as LIBTAYO for cutaneous squamous cell carcinoma. There are a few trials ongoing in phase 2 currently. We are also coming up with a product in phase 1, which is RP2. So in addition to what RP1 was expressing, it additionally expresses anti-CTLA4, which is cytotoxic T lymphocyte associated protein 4. And again, we are trying monotherapy of this product as well as its combination with Opdivo. I'll go over Opdivo and LIBTAYO products in the coming slides. And currently in PD1 non-responsive tumors, we are in preclinical stage for RP3, which expresses additional genes for CD40L and for 1BBL genomes. So Opdivo is a product by Bristol Myers Squibb, and it's nivolumab. It's a human IgG4 monoclonal antibody, which binds to the PD1 receptor and blocks its interaction with PDL1 and PDL2, thereby releasing PD1 pathway-mediated inhibition of immune response, including the anti-tumor immune response. So PDL1 and L2 are expressed on the tumor cells, and these bind to PD1 receptors on T cells. Once they bind, they lead to inhibition of the cytotoxic T cells. And because it acts by removing an immune system checkpoint, it may also cause T cells to attack healthy cells. That's one major disadvantage of this monotherapy just with Opdivo. LIBTAYO is an anti-PD-1 therapy from Regeneron and Sanofi in collaboration. And here we can see that a normal inactive T cell is unable to attack the cancer cell because these can avoid being attacked by T cells by using a defense called the PD-1 pathway or the programmed death 1 pathway. This causes the T cell to become inactive. LIBTAYO can help restore this T cell activity. It blocks the PD-1 pathway, and then hence it's able to make T cells active again so that they can target, attack, or kill the cancer cells. Sometimes, again, these T cells may also recognize and target normal cells. Hence, the product that I was talking about here, RP1, is really helpful in a way for monotherapy because it is not affecting healthy tissue. That's one big major breakthrough with the oncolytic viruses these days. Talking about routes of administration for oncolytic viruses in clinical trials, it has been seen the major routes of administrations have been related to intratumoral injections, followed by intravenous administrations. Other routes have been tried, including hepatic perfusion, intraperitoneal injections, intravesical approaches, or intradermal approaches as well. These were for clinical trials up to the year 2020. So moving on to the process of analytical or the need for analytical development with regards to these vaccines or immuno-oncolytic viruses, I wanted to touch base for certain processes that go on a daily basis, apart from the theoretical aspects of these tumors, how actually these are relevant in the lab and what we as analytical development scientists do to measure these immune responses. One way of handling these is cell-based, plaque-based assays. So we have a cell line, maybe, for example, we use adherent cell lines for Vero cells, and these are adapted from the kidney cells of an African green monkey. These adherent cell lines are infected using different concentrations of virus. So the basis behind these assays would include infection of these cells on a 6 or a 12 well plate platform. And these infection is followed by adding an overlay, which kind of subsides the infection material and kind of binds it to the adherent cells. These are incubated for a few days, followed by removal of this overlay from these plates. This further is, now we can use two approaches wherein we can use primary and secondary antibodies to stain these plaque forming units. And once these plaque forming units are stained, we can enumerate the plaque numbers on these plates. As you can see in the plates below, these plaque numbers are based on the concentration of viral load that was initially infected on these particular plates. So the sequence of events for a normal plaque-based assay would include seeding of cells. So basically, we are seeding these cells at a certain cell number that we need to infect on a 6-well plate. These are followed by infections at different dilutions. Again, usually it's starting from the lowest to the highest dilution in a clockwise fashion. And right after infection and incubation for about 6 hours, we add an overlay medium which contains CMC in combination with DMEM, basic media, which helps to bind these to the adherent cells. And these are left out for between a period of 1 to 3 days for incubation. After that incubation step, these are fixed using either methanol or an ethanol in combination with another fixing media. And once these cells are fixed, they can be stained based on the method developed by the lab. For dengue vaccine candidates, I was using primary, secondary body antibody stainings. We have internal controls that can be used for this particular method, which would stain the plaque forming units based on what virus we are exactly characterizing there. This is followed by counting these plaque forming units usually manually, and then calculating the PFU per ml, which are plaque forming units per ml, and these define the viral load or the viral infectivity in a particular dose of a vaccine or an immuno-oncolytic product. And this is helpful for process development in a way, because once they are sending us in process samples, they want to know the right dose and the amount of virus that they have loaded onto a lyophilized product, in terms so that they are able to develop a better process in terms of number of PFUs they need uh, to get a therapeutic efficacious product at the end. So basic methodologies to successfully conduct these plaque-based assays would include a good aseptic technique, knowledge of routine cell culture under GLP. Very important is attention to detail while running these protocols, because pipetting errors could lead to differences in viral loads, which should be avoided. And observing safety practices while handling this particular virus, be it HSV or I was working with dengue vaccine, Zika vaccine virus, which are live attenuated. So always be use all good PPE and engineering and administrative controls of BSL-2 levels, and obviously disposing these biohazardous material in compliance with local regulations in a safe and proper manner. So these, coming back to these plaques, the ones that are too numerous to count, we can see that on the top plate, the third well, and moving on to fourth, fifth, and sixth well, these should be in a range, and usually it's defined between 10 to 100. Anything below the 10 mark should not be included, and anything above 100 is also not included. So any number of PFU between 10 and 100 is used for calculating the viral load. And we can see that these wells in the top plate are too numerous to count after a point. We can still count the first well. These are placed on light illuminators, so it's easier to count them using a marker. And I believe the third well here is, again, it's lower than 10, so it won't be used for calculation. And sometimes we were able to tell, looking at at this plate, that the viral load in the first well, if it's too high, which means that the log titer that would be converted for the particular vaccine dose or the drug product we receive is obviously higher. And sometimes we would observe a trend wherein we would see the lowest number in the first well going up. Probably in the fifth or the sixth well, we'll see numbers such as 87 or higher, or somewhere around 60, between 60 to 90, wherein the log titer for this product could be lower. But these are visual estimations and come with experience and practice. Other analytical methods that have been employed and are being employed in the analytical development arena include ELISA's plate-based methods for 96 well plate-based DSA, bovine serum albumin impurity detection, specific antigen-based assays. Currently, I'm for HSV antigen, I'm developing a method. There's a method for benzonase. So these would involve identifying a target and then binding a primary and a secondary antibody specific to that particular target. There are certain alpha LISA-based methods for chemoluminescence from newer technologies. Companies such as PerkinElmer have come up with custom-made ELISAs for products within the companies. For example, for Zika vaccine candidate, I was working on something called donor and receptor beads that are custom made for our own products. So they would provide those reagents which encode the antibodies and the antigens that are required for our purposeful targeting in that product. Other methods include reverse phase HPLC for concentration measurements, SDS page for purity determination, gel-based. Sorry. And other ligand binding assays for products such as norovirus vaccine candidates. So future directions for immuno-oncolytic products include, as of now, we have 8 oncolytic viruses in phase 1, 9 in phase 2, and 2 in phase 3 clinical trials as of 2016. And the major part is the ability of these oncolytic viruses for near universal therapeutic impact in cancer, which makes them a unique tool because they are not affecting the healthy cell overall. So we look forward to this new range of therapy that targets only the tumor cells and which further brings a holistic healing approach to patients suffering from cancer. That would be all for today's session in terms of oncolytic viruses. I'm happy to take questions, and just I'm not sure the range of audience here, but for students and master students and above, I'm happy to take questions about your CV, resume preparation, career opportunities, or manuscripts, or you have basic questions on study abroad. You can definitely reach me at my email address here, which is pratik_kakkar@uri.edu. Even if you need any help with revising a manuscript or your resumes for job applications, feel free to reach out for any comments, and I'll be happy to help in those regards. Here are a few pictures that remind me of my 6 years at KLE College of Pharmacy. It was a great experience there, and I would like to thank the college for again inviting me here. I know it's a shorter session. It's actually midnight here, and I was called in for an early shift for the GMP manufacturing tomorrow morning. But I'm happy to take questions, and I really thank Professor Uday Bolmal sir, Dr. Jalal Pure, Professor Rajashri Masaredi ma'am, and Professor Umashri Kokatanur ma'am for all their support for this particular presentation. Thank you all.
Any questions from the delegates? Chat box? No? Sir, I have one question. You mentioned one of the FDA approved analytical product. One of the FDA approved analytical product, oncological product you mentioned. With this product, can we treat the cancer, or only we can enhance or increase the survival rate of the patients?
That's a really good question. Currently, we are working. So in this field, that would be a very big deal to when we say we are treating the cancer. We have different cohorts of patients in the pipeline currently wherein we have seen remission, but we would not be able to comment on long-term efficacy because these are still ongoing clinical trials wherein the patients have been just given the dosage in the past 1 to 2 years. But for sure, the survival rates are increasing. The 5-year survival rates have been seen going up for many of these products, and we hope to see remission, complete remission in all survivors, hopefully, as the research goes on and we are able to get these products out further.
Okay, thank you, sir. Any further questions from the delegate?
No.
Okay, so if no questions, we'll conclude the session. Thank you, sir, for giving a very informative and a very new topic, that is a gene-based cell therapy, where you have touched upon the topics that different gene therapies, that is like CAR-T therapy and virotherapy with oncocytic viruses, and different analytical techniques which are used for these viruses and gene therapy. It was very new topic, gene therapy, and I think all delegates have taken the good opportunity, and it is also a first area we can say. Thank you, sir. With this, I thank you for accepting our invitation in your busy schedule. I know that it is a night time over there. You are giving your presentation at the night time. Thank
Not
you
a
so
problem.
Okay. Thank you so much, sir. Thank you.
Thank you, everyone. Have a great rest of your day and feel free to reach out anytime. I can share my email address.
Wait for 2 minutes. Sanjana ma'am is going to talk to you.
Yeah, sure. I'll share my contact details here. And again, feel free to reach out if you have any questions, any advice you need. I'll be happy to have.
Good morning, Pratik. How are you doing?
Great, ma'am. How are you doing? Good morning.
So very happy to see you on this platform, and you've given good, relevant information. And I'm sure I'll be able to keep up your address, email and everything, so that it will be useful for our students, because this is very important, especially preparing your resume and email. I mean your CV, and to get overseas opportunities for our students. Okay. So thank you so much for accepting. Okay.
Great. It's an honor to be here and good to see you all. I'm not sure if I've met all the students, but I really miss the college and all the professors. It's thanks to all of you I have reached this position, and absolutely feel free to contact me even in the future.
Whenever you come here, make sure you visit the college. We'll be more than happy to see you.
Absolutely. With the current situation, that's a little tricky, but yes, once I'm able to visit, absolutely, I'll be visiting the college.
Okay, carry on. Good day.
Thank you, everyone. Have a good one. Thanks.